All right, and now for the last of the recordings, and this is chapter 21, I guess, bioinformatics and looking at genomes. And this is going to be real generalized. I didn't do everything in this chapter. But there is a new science called bioinformatics, and it's not all that new, but it's basically using computer applications to look at biological data. And so there are degrees now in this. And all this DNA information, which is huge, is now going into these networks, trying to make sense of all this information. And so one of the earlier projects was the Human Genome project, looking at the human genome, trying to map it. And they did map one male. It began in 1990, was completed in 2003, more or less, but I will tell you that it got done a heck of a lot quicker than they expected. I thought it was going to be a 20-year process, and it turned out the reason it was done quicker is because computer technology got so much better, and computers became so much faster. And then the second thing was so many more people were working on it than suspected. So they published in 2006. There was a big controversy on how they should publish it. And at one point people wanted to hold on to it and actually trademark genes. So if you wanted that information, you'd have to buy it from them. But there was another group that said, oh, hell no, this should be public knowledge. And they ended up going ahead and publishing it. And by doing that, everybody pretty much published, because kind of shamed into it, I suppose. Again, we did a male so we could have an X and a Y chromosome. And the number of genes that human genes thought were about between 50 and 100,000. And then that number kept dropping to 40,000, 35, and today we believe it's somewhere around 21,300. And so we have quite a few fewer genes than a lot of other organisms, but we also think it's because that within a single gene, there's alterations like we've talked about in some of the earlier videos. So it's possible to do lots of things with lower amounts of genes. All right, gene annotation. So what we're trying to do is actually kind of mark where things are going and their ultimate function. So the idea now is, with bioinformatics, is not only to read your genes, figure out what those genes are, figure out those sequences, now figure out what those sequences actually do. And so that's a huge part of bioinformatics today. Proteomics is another word you might hear, and this is very similar to what we just talked about, but here we're looking at sets of proteins. So we're looking at what proteins do, where they're found, and again, it's kind of a mapping of how proteins are made, what they do, and so we're really kind of getting at that molecular level of what's going on. All right, and then the last thing I want to talk about is transposons. These are jumping genes. These are pieces of DNA that actually move. So although we have these sequences, we always assume that, it's kind of like this book that always has the same words in the same places, and it turns out, no, that we have genes that actually move, and we have these DNA intermediates that'll kind of be inserted. But transposons are kind of a cut and paste. They'll be cut out of one place and put into another. And by doing that, it can have major effects on how things develop. And so there's an article I want you to read about identical twins and the unique brain. So that will be included in the module. And then not only are there transposons, but retrotransposons, which have RNA intermediates instead of DNA, and it converts DNA, but it does it with reverse transcriptase. So it's actually read in the opposite direction. So it's actually flipping that information. And with this going on, it's going to make it even harder to kind of map and figure out where things are, especially when everything's a moving target. So again, I think the purpose of some of these last lectures is just to get you to realize how complicated all this information really is, and how much we really don't know on what's going on. And that would be the end of